right, let's go ahead and look at photosynthesis. So, the photosynthetic structure that we're looking at in the plant is the leaf. So, you should know at this point that your leaves contain chloroplasts, and the chloroplasts are going to be our site of photosynthesis. So, really quickly, we're going to just look at a general overview of the chloroplast. And I know that we've kind of looked at this when we did our organelles, but we've got our outer membrane, and then we have the inner membrane, and the background that you see right there is going to be our stroma. This is a part of our photosynthesis. We'll see a part of it happening in the stroma. That's going to be the stage 2. And then we've got the main location where we'll see a lot of things happening are our thylakoids. And if we looked at the entire stack of the thylakoids, that would then be called our granule. So keep in mind, thylakoid is each individual disc that you see in the stack. I have our reaction written out at the top. If y'all remember, we looked at cellular respiration and we said go cow, but for photosynthesis it's cow go. You've got your CO2, H2O, plus light will give you glucose and oxygen. So when we're looking at the first part, we're looking at the thylakoid membrane. All right, so we're going to say that we're going to zoom in into the membrane of the thylakoid in a little bit. Before we go in and look at the thylakoid membrane, I just want to kind of do a quick review about the chloroplast. So chloroplast is your site of photosynthesis. This is where we're going to have all of the processes of photosynthesis happening. And overall, we will have 2 stages of photosynthesis. Stage 1 that we're going to see, that's what's going to happen in the thylakoid membrane. Okay, so stage 1 will be in the thylakoid membrane, and that's the light dependent reactions. And then stage 2 will then be in the stroma. Okay, so stage 1, that's your light dependent reactions. And stage 2, that's your light independent. So we do not need light in order for stage 2 to occur, and that was that common misconception that plants can only do photosynthesis when they are exposed to light. But a part of photosynthesis is actually light independent, so it can still continue without light as well. So stage 1's in the thylakoid membrane, stage 2 is in the stroma. Alright, so when we start looking at the thylakoid membrane, one of the components that you need to remember is going to be chlorophyll. Alright, so chlorophyll is a big component of the thylakoid membrane, and this is the photosynthetic pigment. This gives plants the staple green color. Okay, this is the colored substance that's actually absorbing the light energy. Okay, so this right here is going to be a very big component of stage 1, and we will find chlorophyll in the thylakoid membrane as a part of the proteins. All right, so chlorophyll is attached to the proteins in the thylakoid membrane. So when we were looking at PS2 and when we're looking at PS1, a part of the PS2 and PS1 structure is chlorophyll. And so the proteins in the chlorophyll that are a part of the thylakoid membrane, they make up the photosystem. Okay, so when we're referring to the photosystem, you've got to keep in mind that we're referring to the proteins plus the chlorophyll. All right, so the process that we're going to have in stage 1, we're going to start off with light. So light is going to hit photosystem 2. So remember that photosystem 2 is actually going to come in first. Photosystem 1 comes in later. It's a common misconception. They were not named according to the order that they occur in. They were named according to the order that they were discovered. So since photosystem 1 was discovered first, it got photosystem 1, and this got photosystem 2. But it happens first. All right, so the very first step that we're going to have is that the chlorophyll in photosystem 2 is going to absorb light energy from the sun or a light source. Now, the energy that it's absorbing is going to excite the electrons in the chlorophyll to a higher energy level. Now, this process, when these electrons are moving to the higher energy level or changing to the excited stage, is known as photoionization. So what we're going to see is that these high energy electrons, they're going to leave the chlorophyll in photosystem 2, and they're going to travel along through the thylakoid membrane along a series of carrier proteins. So they'll move from photosystem 2 along the membrane over to the next carrier protein, onto the next carrier protein, onto the next carrier protein, and on to photosystem 1, and so on. Now, as they are moving through these carrier proteins, they're leaving a little bit of energy behind after each of the moves. So each transfer of the electron releases a little bit of energy that's left behind for those proteins to use. All right, so remember when we learned about our cell transport, our protein pumps for active transport need energy. So this is the energy that they're then going to use to pump hydrogen ions that are in the stroma, and then they will pump those hydrogen ions into the thylakoid. All right, so this entire process is kind of all working hand in hand. So electrons, as they're moving through, transferring from one carrier protein to the next until they reach photosystem 1, and even on from photosynthesis 1, are leaving behind a little bit of that energy, and then that energy is then being used by the pumps. Okay, so it's actually the energy that is needed for our thylakoids to complete their functions. All right, so next what we're going to see is these hydrogen ions that keep accumulating inside of our thylakoids. We're going to see a similar process to what we saw in cellular respiration. After a certain amount of time, when we keep on accumulating hydrogen ions, we will end up leading to the process of diffusion, where our hydrogen ions will move from an area of high concentration to an area of low concentration. But before we go into actually looking at that, I want to kind of reiterate the active transport. So our hydrogen ions are moving from an area of low concentration to an area of high concentration. So this is active transport, and the energy that we're getting is the energy that was left behind by our electrons. So I know it's a little repetitive. I just want to make sure that we can make that connection from what we learned in Unit 2. Another way that we are adding on to the hydrogen inside the thylakoid is actually with water. So we know that we have to keep watering plants. Now, that water is in the thylakoids. Those water molecules within the thylakoids are going to be broken down. And as those water molecules are broken down, each part of the water molecule will go somewhere different. Hydrogens will end up staying and adding on to the hydrogen ions that are within the thylakoid, which will eventually diffuse. The electrons from them breaking down will actually go in and replenish photosystem 2, because that photosystem 2, the electrons have moved on. They got excited, they moved on and traveled through the carrier proteins. So they will need to be replenished so that it can continue working. And hydrogen adds on to the accumulation of hydrogen, and the oxygen will actually be released. That's the oxygen that we see in our reaction for photosynthesis. the oxygen is being released in this part of photosynthesis in stage 1. All right, so let's talk about photosystem 1. Okay, so we know that the light's hitting photosystem 2, and as it's hitting photosystem 2, it's starting that process of exciting the electrons, causing them to move along. Well, the light, at the same time that it hits photosystem 2, it's going to be hitting photosystem 1 as well. And so, similar to photosystem 2, this will excite electrons and continue on that electron transport chain. So we have chlorophyll in photosystem 1 as well. We'll have that same process. The chlorophyll, the electrons will get excited and then they'll end up continuing to move on in photosystem 1, just like photosystem 2. Now, this would be important in continuing our chain. And now let's go ahead and talk about those hydrogens. So these hydrogens that we accumulated, these hydrogens, now that we have an area of high concentration for our hydrogen ions, once they've accumulated, they are going to diffuse through ATP synthase. Okay, so very similar to when we did cellular respiration. We're going to think of this as like a rotor as the hydrogen ions move through the ATP synthase. It's called ATP synthase because it's synthesizing ATP. The movement of the hydrogen as it turns that rotor helps to bring together our ADP and a phosphate, creating ATP. All right, so it's very similar to cellular respiration, and that's kind of why we went into a lot of detail with cellular respiration, because we're going to see a lot of similarities between photosynthesis and cellular respiration. So you'll see the hydrogen ions as they diffuse through, they'll turn that rotor of ATP synthase, which will help bond ADP to a phosphate, releasing or creating ATP. And as y'all remember, we learned about ATP, that's your phosphorylation, but we're going to call it photophosphorylation because this is happening as a part of photosynthesis. Okay, so keep in mind this is our photophosphorylation. All right, so we've looked at the creation of ATP. The other thing that we need to look at is going to be the production of NADPH. So if we come back to looking at how our electrons are moving through our transport chain, okay, so we need to know that while they're moving through this transport chain, one of the other processes that's happening at the same time is that these electrons and the energy from these electrons is helping to bond NADP plus and hydrogen ions to create NADPH, which essentially is going to serve as our hydrogen ion carrier in photosynthesis. All right, so this creation of NADPH and also the creation of ATP, both of these are actually going to come in handy when we look at stage 2. All right, so all of this that we just looked at, this was stage 1, our light dependent reactions. Okay, so stage 1 is a light dependent, and what we ended up creating was our ATP and NADPH, and we made oxygen. So now we'll go on to stage 2. This is our light independent reaction. So we're really going to take that NADPH and we're going to take ATP and we're going to continue on the process, and it's going to be happening in the stroma. All right, so this is the continuation from stage 1, where we can now finally make glucose. All right, so the process in stage 2 will start out with a 5-carbon molecule that you just need to know RUBP. This, even though we're going to call this a 5-carbon molecule, remember that it doesn't mean that it's just those 5 carbons. We have other molecules as well. So this is the actual formula for it would be C5H12O11P2. So the process with this 5-carbon molecule will start when carbon dioxide that's diffusing into the stroma. That carbon dioxide will diffuse into the stroma. It will react with RUBP, creating a 6-carbon molecule. All right, so that carbon dioxide attaches to this 5-carbon molecule, creating, which is now a 6-carbon molecule. And this process that we're seeing with the carbon dioxide reacting is also known as carbon fixation. All right, so now that we now have a 6-carbon molecule, this is where our NADPH and ATP will come in. All right, so the ATP and the NADPH are going to help break this 6-carbon molecule down into into 2 3-carbon molecules. All right, so we end up absorbing the hydrogen from the NADPH, which will then release the NADP plus, and we'll end up using the energy as we break ATP down into ADP. All right, so the next step, we've got enzymes, we've got ATP, we've got NADPH. All of them are going to be working together to break a 6-carbon molecule that we have right here down into 2 3-carbon molecules. Now, even though this right here is a 6-carbon molecule, this 6-carbon molecule is not glucose. All right, so we have to keep that in mind. This is an intermediate until we get to glucose. So we release NADP plus, we release ADP, both of which will be reused and recycled later. So now we've got our 3-carbon molecules, which are also known as PGAs. Now this process right here is continuously happening, and it's happening with multiple molecules of RUBP. So we have to realize that this process is going to be repeating, and we're going to continue to create these PGAs, which are also known as phosphoglycerates. Now, the next step from right here, we can have one of 2 things. One of the things that we can see is that now we can take these 3-carbon molecules, and some of them will end up bonding, and that's where we would make our glucose. So some of these 3-carbon molecules will bond to make a 6-carbon molecule, and that 6-carbon molecule is going to be glucose. All right, so this is where the glucose is being produced in photosynthesis. The other option, what some of the other PGAs will do, is that those 3-carbon molecules will end up keeping the cycle going. So those 3-carbon molecules, with the help of ATP, will be broken down further to help us, continue on the cycle and get back to that 5-carbon molecule. So ATP molecules will break down several of the PGAs. Now that those carbon molecules are completely broken down, with the help of some enzymes, we will end up reforming RUBP. All right, so ATP is breaking our 3-carbon molecules down, and then enzymes help to make RUBP, and the cycle continues. We'll end up having carbon fixation again, break our 6-carbon down into 2 3-carbon molecules. Some of them go on to make glucose, some of them go on to continue our cycle. All right, so do y'all see how similar this is to the Krebs cycle? It is a lot less steps than the Krebs cycle, but it is very similar. So overall, CO2 plus ATP plus NADPH were all used in stage 2 to create glucose. All right, so all of this, so CO2, ATP, NADPH, and them combining to create glucose is kind of a quick summary of our light independent reaction. So I'm going to kind of look at overall, let's go back and look at our reaction. So the CO2 in our reaction is coming from stage 2. That was the CO2 that goes in stage 2 to start up our little cycle. We see our H2O, which is our other reactant. We see that in stage 1. So if y'all look at, remember when we saw that H2O molecules in stage 1, inside the thylakoids, how they broke down, adding on their hydrogens to the accumulation of the hydrogen ions. They gave their electrons to continue to replenish photosystem 2, and they released oxygen. So that's where our H2O is. The glucose in the reaction, which is one of our products, is produced in stage 2. So that's the main product that we're getting out of stage 2. Okay, so the last thing we have left are oxygen in the reaction, as we just looked at, like from when we broke down our H2O. So the oxygen in that reaction is coming from stage 1. That's the oxygen that then gets released as waste in stage 1. Okay, so when those water molecules break down, the oxygen that's left, that's released as waste. All right, so that's like a quick overview of where everything in our photosynthesis reaction is.